All right, buckle up, everyone. We're diving deep into the world of neurotransmitters today.
Specifically, we'll be focusing on two of the most important players in your central nervous system.
GABA and glutamate.
You've given us a really interesting set of sources on these neurotransmitters, and
Yeah, it looks like you're really looking for a unified understanding of how these systems work.
absolutely, and we're going to try to break that down for you.
So we want to help you identify all the key players.
Different receptor types and
Their roles.
even how imbalances in these systems can lead to conditions like epilepsy.
I mean, these are really essential for understanding how the brain works.
They really are.
So,
Yeah.
should we just jump right in?
Yeah, let's do it.
Okay, great.
Think of GABA as the brain's brake pedal and glutamate as the gas pedal.
Oh.
We'll explore how they work together to keep your brain functioning smoothly.
So I always hear about GABA being inhibitory,
Right.
but one of the sources points out that glycine actually takes the lead in the spinal cord. Why is that?
That's a great observation, and it's a really important distinction.
Okay.
While GABA is the main inhibitory neurotransmitter in the brain, glycine steps in to keep things calm in the spinal cord.
Gotcha.
Imagine, for example, motor neurons signaling muscles to contract.
Okay.
Without inhibitory signals, those muscles would be constantly activated.
Oh, wow.
It would be exhausting.
Yeah, I can imagine.
That's where glycine comes in. It acts on glycine receptors located on those motor neurons.
Okay.
And when glycine binds,
Yeah.
it opens the receptor, leading to inhibition of the motor neuron
Okay.
and preventing uncontrolled muscle contraction.
So it's like a carefully calibrated braking system.
Exactly.
Okay, that makes sense.
It does.
But this brings up a question from another source.
Okay.
How can different receptors on the same cell, both activating the same cascade, lead to different effects? It's like getting two different outcomes from pushing the same button.
That's a great question, and it really gets at the heart of how cells achieve specificity in signaling.
Okay.
It turns out that cells are much more spatially organized than we once thought.
Okay.
The source uses the example of cardiomyocytes, or heart muscle cells.
Okay.
Both beta-1 adrenergic receptors and EP4 receptors on these cells
Yeah.
activate the same GABA cascade,
Okay.
leading to increased cyclic AMP.
Yeah, but if I'm reading this correctly, only stimulating the beta-1 receptor actually increases the force of the heart muscle contraction, right?
That's exactly right.
Yeah.
And that really highlights the importance of something called signaling microdomains or compartments. Even though cyclic AMP can diffuse throughout the cell, the location where it's produced can determine its specific effect.
So it's like having different departments within a company.
Yeah.
The same memo might be sent to everyone,
Right.
but the impact will vary depending on who receives it.
A perfect analogy.
And this is where A kinase anchoring proteins, or AKPs, come into play.
Achy caps, yes.
These proteins act like scaffolds, localizing specific signaling molecules to particular areas within the cell.
That's right.
So AKPs are like the traffic controllers of the cell, directing signaling molecules to their designated locations.
Exactly.
So it's not just a random scattering of signaling molecules. It's a very organized system.
It is.
With specific players being directed to specific locations.
Precisely.
And this level of organization is crucial for ensuring that signals are interpreted correctly
It is.
and lead to the appropriate cellular responses.
Absolutely.
This makes me think back to GABA and glycine.
Yeah.
They're both inhibitory,
Right.
but they have different roles in different parts of the nervous system.
Yeah.
Is this a similar concept of location-determining function?
It absolutely is.
Okay.
In fact, the receptor subtypes for GABA also contribute to the specificity.
Okay.
We have two main types of GABA receptors, GABA and GABA.
Okay, now this is where things start to get really complex. Can you break down the differences between these GABA receptors for us?
Certainly.
Okay.
GABA receptors are ionotropic receptors, meaning they directly control the flow of ions across the cell membrane.
Okay.
When GABA binds to a GABA receptor, it causes the channel to open,
Yeah.
allowing chloride ions to flow into the neuron.
Okay.
This makes the neuron more negative, making it less likely to fire an action potential.
So it's like GABA receptors are putting the brakes on neuronal excitation very directly.
Exactly.
Oh.
GABA receptors, on the other hand, are metabotropic receptors.
Okay.
This means they work indirectly through a series of intracellular signaling steps.
Okay, that's a bit more complicated.
A little bit.
What happens when GABA binds to a GABA receptor?
Well, it sets off cascade of events that ultimately lead to the opening of potassium channels.
Okay.
Potassium ions then flow out of the neuron, again making it less likely to fire an action potential.
So both GABA and GABA receptors ultimately inhibit neuronal firing.
They do.
But they do it through different mechanisms.
Precisely.
And this difference in mechanisms is important because it means that drugs targeting these receptors can have distinct effects.
That's a very good point.
This is reminding me of how thyroid hormone receptors can also have different effects depending on their type and location.
That's right.
One of the sources dives into this, highlighting how they can even act as silencers or activators of gene expression.
That's a fascinating example
It is.
of how a single molecule can have diverse effects depending on the context. In the absence of thyroid hormone, or T3, they bind to DNA and repress gene expression.
Right.
But when T3 binds, they undergo a conformational change and activate gene expression.
Like a two-way switch.
It is.
And to add another layer of complexity, the type of DNA sequence they bind to can also influence their function.
That's right.
Sometimes even leading to gene repression in the presence of T3. It's like having a key that fits into different locks.
Yeah.
And produces different effects depending on the lock it's in.
A brilliant analogy.
And to make things even more intricate, TRs can also form homodimers or heterodimers with other nuclear receptors.
Right.
Further expanding their regulatory network.
That's right.
It's like a beautifully choreographed dance where the partners and their movements determine the overall outcome.
I like that analogy. I feel like we're just scratching the surface of these complex systems.
We are.
We've seen how location and receptor type can lead to very specific effects for both inhibitory and excitatory signals.
We have.
But where does glutamate, the gas pedal, fit into all of this?
That's a great question, and it's something we should definitely explore further.
Okay.
Glutamate also has a variety of receptor subtypes, each with its own unique role in modulating neuronal activity.
Okay.
But perhaps we should delve into that in more detail in the next part of our deep dive.
Okay, good.
Okay, so let's shift gears a bit.
And rev up those engines and talk about glutamate, the excitatory counterpart to GABA.
Yes. Let's explore that gas pedal of the brain.
Right, so
one of the sources dives into glutamate receptors, and it seems like there's a whole family of them, just like with GABA.
That's right.
Okay. So
Glutamate receptors come in two main flavors.
Okay.
We have ionotropic and metabotropic.
Okay, I remember those terms from our GABA discussion. Ionotropic receptors are like the direct acting ones, like a light switch.
Yes.
And then metabotropic receptors are the more indirect ones, like a dimmer switch with multiple steps.
Exactly.
Okay.
So let's talk about the ionotropic glutamate receptors first.
Okay.
There are three main subtypes, and they're named after their specific agonists.
Okay.
AMPA, NMDA, and kainate receptors.
So different molecules can activate different types of glutamate receptors.
That's right.
Interesting. What's the significance of that?
Well, it's all about, fine-tuning neuronal excitation.
Okay.
AMPA receptors are kind of the workhorses of excitatory signaling.
Yep.
They're responsible for fast, short-lived responses.
Right.
When glutamate binds to an AMPA receptor, it causes a rapid influx of sodium ions into the neuron, depolarizing it, making it more likely to fire an action potential.
So NMDA receptors are like the quick burst of energy that gets things going?
That's a good way to put it.
Okay, what about NMDA receptors? They sound a little more complex.
NMDA receptors are definitely more complex.
Okay.
And they play a crucial role in something called synaptic plasticity, which is the ability of synapses to strengthen or weaken over time.
Wow, so these receptors are involved in making our brains more adaptable?
They are.
That's incredible.
It is pretty amazing.
So what makes them so special?
Well, NMDA receptors are unique in a couple of ways.
Okay.
Firstly, they require both glutamate binding and membrane depolarization to open. This means they only become fully active when the neuron is already somewhat excited.
Oh, so it's like a safety mechanism preventing excessive excitation.
Precisely.
Wow, okay, and what's the second way?
Secondly, when NMDA receptors do open, they allow calcium ions to flow into the neuron.
Okay.
And calcium acts as a second messenger, triggering a cascade of signaling events that can lead to long-lasting changes in synaptic strength.
So NMDA receptors are not just about immediate excitation, but also about shaping the brain's connections over time.
Exactly.
Wow, okay.
So we have AMPA receptors, NMDA receptors, and then we have kainate receptors.
Okay.
Now, kainate receptors are, they're less well understood than AMPA and NMDA receptors,
Okay.
but they seem to play a role in modulating neuronal excitability as well.
So it sounds like glutamate signaling is this carefully orchestrated symphony of different receptor subtypes, each contributing its own unique melody to the overall neuronal activity.
That's a beautiful analogy.
And just as with GABA receptors, different drugs can target specific glutamate receptor subtypes.
That's right.
Allowing for more fine-tuned pharmacological interventions.
Exactly.
This makes me wonder, if both GABA and glutamate are involved in regulating neuronal activity, how do they work together to maintain balance in the brain? One of the sources mentions something called synaptic integration.
Right, synaptic integration.
Yeah.
So synaptic integration is the process by which a neuron sums up all the excitatory and inhibitory inputs it receives and decides whether or not to fire an action potential.
So it's like a constant tug-of-war between GABA and glutamate.
Yeah.
With the neuron acting as the judge.
Exactly.
And when this balance is disrupted, things can go wrong.
They can.
One of the sources discusses how imbalances in GABA and glutamate signaling can contribute to epilepsy.
That's right.
And epilepsy is a serious neurological disorder.
It is.
Characterized by seizures.
Yeah.
So how does this imbalance play a role?
Well, in epilepsy, there's often an overexcitation of neurons in certain brain regions. And this can be due to a variety of factors, including a decrease in GABAergic inhibition or an increase in glutamatergic excitation.
So either the brakes are failing or the gas pedal is stuck.
That's a great way to visualize it.
Oh.
And this excessive neuronal firing can spread to other brain areas, leading to the characteristic seizures seen in epilepsy.
So this is where understanding the specific receptor subtypes becomes really important, right?
Absolutely.
Okay.
By targeting specific subtypes of GABA or glutamate receptors, researchers are developing drugs that can help to restore balance and reduce seizure activity.
That makes sense.
It does.
This brings me to another point from the sources.
Okay.
One of them mentions that glycine isn't just about inhibiting motor neurons in the spinal cord.
Right.
It actually modulates NMDA receptors in the brain as well.
That's a good point.
Yeah.
Glycine acts as a coagonist at NMDA receptors, meaning that both glutamate and glycine need to bind for the receptor to fully open.
Wow, okay.
So this adds another layer of complexity to the interplay between these neurotransmitter systems.
It's incredible how all these different molecules and receptors are intertwined.
It is.
Constantly interacting to fine-tune brain activity.
It's a truly remarkable system.
It is.
And we haven't even touched on the metabotropic glutamate receptors yet.
Oh, boy.
They add another dimension to this complex interplay.
Okay.
But perhaps we should save that discussion for the next part of our deep dive.
Okay, let's shift gears and delve into those metabotropic glutamate receptors next.
Okay.
Okay, so let's jump into those metabotropic glutamate receptors.
All right. Sounds good.
We've covered the inotropic receptors for both GABA and glutamate.
Right.
But you mentioned metabotropic glutamate receptors. I'm curious to learn how those fit into this intricate puzzle.
Yeah, metabotropic glutamate receptors, or MGLORs for short, add a whole new layer of complexity to glutamate signaling. Unlike their ionotropic counterparts, which directly control ion channels, MGLORs work indirectly through a cascade of intracellular
command rather than issuing direct orders.
Exactly.
Okay.
And there are 8 different subtypes of MGLORs, grouped into 3 main families based on their structure and function.
Okay.
Some of these receptors are located on the presynaptic neuron, while others are found on the postsynaptic neuron.
Okay, so this is where my head starts to spin a little bit.
It can get complicated.
Can you give us an example of how these MGLORs actually work in the brain?
Sure.
Yeah.
Let's consider mGLR2, which is located on the presynaptic neuron.
Okay.
When glutamate binds to this receptor, it triggers a signaling cascade that ultimately inhibits the release of more glutamate from the presynaptic terminal.
So it's like a feedback mechanism, right? When glutamate levels get too high, mGLR2 steps in and says, okay, that's enough excitation for now.
Precisely.
Yeah.
This type of negative feedback loop helps to prevent excessive excitation and maintain that balance in the brain.
Gotcha.
Now, other embryors
Yep.
located on the postsynaptic neuron
Okay.
can modulate the activity of ion channels,
Mm-hmm.
influencing the neuron's excitability
Okay.
and its response to other neurotransmitters.
So it sounds like MGLRs are playing a more subtle but equally important role in fine-tuning neuronal activity.
I would agree with that.
Okay, that makes sense.
And because of their diverse signaling pathways and locations,
Yeah.
MGLRs represent very promising targets for drug development.
Right.
Researchers are exploring their potential for treating a wide range of conditions,
Okay.
including anxiety, depression,
Wow.
and even neurodegenerative diseases like Alzheimer's.
It's incredible how these tiny molecules, the receptors, can have such profound effects on our brains and our overall well-being.
I know, it's amazing, isn't it?
It really is.
And as we continue to unravel the complexities of these neurotransmitter systems, we gain a deeper understanding of how our brains work and how we can develop more effective treatments for neurological disorders.
That's really exciting.
It is.
So this deep dive has been a really eye-opening journey into the world of GABA and glutamate.
Yeah.
We've learned about their different receptor types, their roles in regulating neuronal activity, and how imbalances in these systems can contribute to conditions like epilepsy.
We've also seen how these neurotransmitters don't work in isolation, but rather engage in a constant and intricate dance, influencing each other and other signaling pathways to maintain balance in the brain.
It's amazing how it all works together.
It really is.
Well, it's clear that there's still much more to discover.
Oh, absolutely.
But I think armed with this knowledge, you're well equipped to continue exploring this fascinating field on your own.
I think so, too.
Who knows what exciting new insights await. Indeed.
So, thank you for joining us on this incredible journey into the inner workings of the brain.
It was my pleasure.
Until next time, keep those minds curious and those questions coming.